Leidy Laura Arias Martinez and PhD students at University of and this project was contributed by myself and my undergrad student colleague, and it was with the supervision of the. So I'm going to quickly talk about the project of Chromosome in an active genus in Triodanis. So the genus belongs to the Triodanis family, is native to North America, it's highly concentrated in America, but we also can find some populations in Central America and a few more in South America. So we have 7 species in the genus, by flora, Triodanis, Triodanis sperma, Triodanis parva, Oscillarius, salmon, or rodents. However, you can find some signs in the literature that Triodanis by flora is a subspecies of Triodanis, but in this case, we're going to use Triodanis as a species. So in England, we have 3 species of Triodanis. So the widely spread Triodanis by is yes. So we probably some people here can see them today in the time. We couldn't see any flowers, but we saw the vegetation. So this is why the other species of the flora is widely dispersed too. These 2 species often coexist and overlapping this distribution, and sometimes we find some hybrids between these 2 species. And in those, we can also find the Triodanis species, as you can see, 3 of them are pretty similar, respectively speaking. However, we can distinguish if we see closely the capsule, this is the fruit, and we see the position of the pore, we can distinguish. So you can see in back waters and with the other species, they look pretty similar, right? So we're talking about a genus that is split. The vegetative and the reproductive traits are very similar, so it's pretty hard to identify. So the pretty morphology and frequent hybridization, well, there's a challenge for taxonomy approach. So that's why we want to explore other kinds of traits that can help us to actually identify species in the genes. So in terms of cytogenetic studies, that is usually the main part, studying the cells, basically the study of chromosomes of plants, that is almost not safe studies for these genes. However, we found 2 papers that mention the number of chromosomes. In this case, in 1983, Love mentioned that for Triodanis by flora, we have an applied number of 30 chromosomes. And in this paper from 1966, Galena just mentioned the diploid number of chromosomes for Triodanis by flora and Perculiana, and they mentioned that the diploid number for that species is 56. So as you can see, it's a difference between these 2 records. We don't know actually why it's the difference between the number of chromosomes. We don't have a lot of information in these papers to contrast why they are similar. So we need to understand more of the topic. So given that the taxonomical context in this genus, inconsistency in literature in terms of cytogenetic studies, we say that the cytogenetic studies are highly recommended and highly needed for the genes. And that's pretty important because chromosome numbers are fundamental to understand the speciation process and hybridization in species. So we found this gap in the knowledge and we want to address by looking at the number of chromosomes. So for that, we growing several species of Triodanis, taking for the calculation for the program laboratory. That's it was collecting previous years in our work. We brought in the greenhouse condition of plant biology greenhouse from spring 2024 to spring to this spring, and we found that 6 to 10 weeks was enough to start to collect the tissue that we want to use for chromosome. So in this case, we use the apical meristem of the plant. So as you can see, it's pretty tiny. We use that book there. We use the tissue to form the chromosome. So we collect the tissue, we use a reagent that we call the killer just to stop the mitosis process and be able to form the chromosomes. After that reagent, we just treat the tissue in full conditions. And when we are ready to actually do the process to form chromosome, we use acid fluoride to solve the tissue and be able to do the next process. So we use the expression method. So it basically just make pressure in the tissue and destroy the cells and spread the chromosome to be able to function. Except for that, we use for stain to be able to look the chromosome in the microscope. After some time in that stain, we proceed for a vertical pressure in the tissue. We can use the drum, like we see in this figure, or we can use they compare to the rest. After that, we just look in the microscope, and say, well, so after a lot of samples, we found that difficulty, of course, we have difficulty in the process. For example, we have different aspects of the mountain process because it's like a physical, we need to have practice and be able to have a good spread. So in some cases, we found that the sister chromatids separated of the chromosome, they look like two chromosomes, but they're actually a sister chromosomes. Other aspects was the lack of chromosome spread, and you can see in fact 2A. So in this case, the chromosome animal spread as well as in 2B. The most common aspect of all the process was the poor spread of chromosomes. So, for example, in this cell, we see for good spread chromosomes, but in this case, the two chromosomes are close, sometimes they overlap. So that's why we need to do a lot of the slides to be able to find a good spread chromosome that you can actually tell. So after a lot of process, we finally find a good spread for some species. So we found out for the Venezuela flora, the dipole number is 28, and for Perfoliata and Leptosperma, we found a dipole number of 30 grams. For the other species of the land, we also found it. We also measure the chromosomes. For now, we just randomly pick 5 chromosomes and 5 cells, and just estimate a mean. So we found similar chromosome size in all the species of Triodanis, with main values for 4.84 micrometer in Perfoliata and until 1.22 in Leptosperma. So pretty small chromosomes, so you can contact with other species as always. Yeah. And finally, thank you to Foreigners National Society for this opportunity of run to be able to fill this gap in the knowledge of these entire genus. This is my student here. And we'll also say thank you to Clean from the plant like the greenhouse that helps a lot with the growing process of the plants. So I didn't know that the questions are at the end, so just thank you.